I think I'm gonna go change to my shirt. I feel bad. Okay, I'm running back.
Not that you don't care.
Gonna change into what?
Our Team Tritonia shirts.
Oh, that's beautiful. Where'd you get those?
There's a website called Circuiter.
Oh
And they
man,
do everything. They have
that's beautiful.
tunicats and sea urchins and bacteriophages, and they're all like top hats and monocles. Jim's got one in his messenger bag.
Who has the shiniest teeth? I think Alexis's.
Yeah. And Elska is pretty good. Alexis, yeah.
You know, not gonna lie, but mine are pretty shiny in there too.
Just laughing at Catherine and
Oh,
it's pretty good.
Birds of a feather. I can't tell if it's working.
Yeah.
Here, watch. Help yourself to a drink if you'd like.
Really?
Why it says help yourself?
I was sure that was for everyone.
That's for everyone.
Thank you.
Even a towel to dry off.
Well, I think it's going. It says it's going, but there's one viewer. So it must be going. I wish that I could somehow view it, but I think that because I'm signed in as me, Stacy Farina is the person who's going because she just sent me a text saying, it's working.
Are you live streaming?
Yes. Yeah, I'm live streaming it, and then the 2 sections will go up on YouTube. So once it's done, you can share them. We're live streaming all the presentations. So,
Are you filming it on that laptop?
Yeah.
Okay. Looking for a key.
no.
Can I search the link right now?
Yes, if you're on Twitter, I've got it.
Oh, I'm not. I'll find it.
Okay, it's on the Facebook page.
Okay.
REU.
Look down. Everything. Did you do that?
Help yourself to beverages.
You got it. You did hear it, sure. Excellent.
Made it too.
That's why they call it try.
It's my only formal wear for the summer. If you can even call this formal.
I like how you say that as I walk in with my.
Yeah, I know that.
I have practiced that, so. No. Oh, yes, I do.
Hey, Scott, let's go.
I know what you. Good. Broadcasting. Right. Very good. Just broadcasted your belly as well.
10 seconds. We won't get chicken for days out.
Oh,
We'll have to sell KFC.
I think this is the first day that everyone's so hungry.
So the nice thing
Come on. I think that's the sensation of having a pee when you don't. Nervousness. It's like, I think I gotta go.
We also smile face.
Did you get a sleep last night? You're not. Did you still wake up at 2 in the morning? Okay. Yeah.
You know, I picked out the 3-hour space one. I think we should go with Tess' choice for the Tritonia. Nobody's good.
Well, that's a bunch of different colors of the.
Yeah. Plus, that's like cheap Tritonia. It's always fun.
Yeah, but, Oh my God.
You know what?
I didn't see the afternoon, so I still, I have a power.
Yeah, you can give it to me after the session.
I was hanging out with them, I'm like, they'll probably come around, right?
Well, it's there for the laptop. So, yeah. The bulk of the infant that I just need one, I don't want to get. I know I can just do 345 and you get and try and get down below. So yeah, no problem at all. So yeah, after this.
Yes.
I ended up drawing the blue lines and then going into Illustrator and straightening them along the points. So I'll figure it out, but right now.
That's a solution. I'm not going to say it's a good solution, but it's a solution. Yeah.
I mean,
Yeah, there's just no way that log curves can curve. Log up.
After this.
As long as that's the one.
I'm really impressed by that.
That happens. Yeah, but that would be a very big math. Logarithms are broken into that would be impressive. You see the conversion of
distinct lack of fish stages everywhere.
Summertime is having fun.
In response to shiny object being pointed, the jaws open.
Is it? Yes.
It is significant.
I mean, that's not serious.
My friends
Nice guy. Same thing.
I didn't use that.
Yeah.
Yeah, I just wanted to get All right, everyone, welcome to the 2015 Links REU Beacon Symposium here at the lab, at Friday Harbor Labs. Woo. Undergraduate research has been a hallmark of life here at Friday Harbor Labs for a long time. It's worth noting at this point, John Blinks, about 20 years ago, it kind of established kind of the first endowment to fund undergraduate research here. And for those of you who don't know, John Blinks was a professor. He was a medical doctor, and he actually pioneered the use of aquarin, a protein found in the jellyfish off the docks here that fluoresces in the presence of calcium ions. And so that has been used to kind of understand what's going on in the heart, and has been used in kind of treatments for heart disease and things like that. And he was the one that in 1996 established an endowment in his mother's honor to support undergraduate research here. And then to build on that, about 5 years ago now, Adam Summers and Sophie George wrote a grant for NSF, and NSF now supports the REU program here. So we were able to expand on that. And this year we have 15 students from all over the country. I think 12 different states are represented institutionally among these students. And really, it's really kind of an amazing group, and you'll hear about all their research today. It's really quite a fabulous program in terms of the mentor and student relationship, and you'll see the fruits of their labors today. So without any further ado, the first speaker today is Teris Oglesby. She's from Savannah State University, and her mentor is Jim Murray. Yeah, that's fine.
Hi, my name is Teris Oglesby, and this summer I was looking at how water flow interacts with the rhinophores in Tritonia tetraquetra, which is formerly known as Tritonia Jimenez. So first, I would like to talk about why studying Tritonia. Tritonia is a model organism for understanding the neural basis of behavior, because these animals have a relatively simple nervous system with large identifiable neurons that with large identifiable neurons that can be easily stimulated and recorded. And here is a picture of a Tritonia brain here. And additionally, Tritonia have simple but limited behavioral responses to environmental cues that can be easily seen in. So not only are these animals very cute, but they provide a great deal of knowledge to neuroethologists who are trying to understand how brain controls behavior. So moving on to my introduction, Tritonia are primarily dependent on odor-based navigation. These animals live in a high Reynolds environment, which is characterized by turbulent water flow. So due to the turbulent nature of the environment, these animals primarily depend on odor plumes to navigate throughout their environment. And here I have a diagram depicting the nature of an odor plume. So as an odor plume washes over an animal, the animal will encounter areas where odor is present, which is symbolized by the shaded areas here, which will then be followed by it encountering areas where there is little to no odor present. And this causes a temporal pattern of odor being present, absent as the animal is navigating throughout the odor plume. And that can be seen here where the odor is being present, non-present, present, non-present, and present. So as an animal as Tritonia is navigating throughout the odor plume, it will crawl upstream in the presence of attractive odors such as conspecifics and prey, and this is known as odor-gated rheotaxis. So during odor-based navigation, the Tritonia has two sensory organs that it uses. The first is the oral veil, which is this structure here, and this structure is involved with detecting water flow as well as involved in activities such as touch and taste. And it also has rhinophores, which are these structures here, which are involved with odor detection. Okay, so for my project, I was interested in looking at how a boundary layer forms around the rhinophore and Tritonia, because we know that a boundary layer will dictate how fast this animal responds to odor changes in the water. So a boundary layer is a thin layer of fluid that sticks to and is carried along the surface of an object or a body. So here I have a diagram of a boundary layer forming around an object. So just pretend that this is a Tritonia body, and these arrows are indicating the speed of water flow. And as water is interacting with the surface area of the animal, the water will slow down and form a boundary layer over the animal. So kind of bringing this back to my project, so if a thick boundary layer forms around the rhinophore, it's expected for the animal to have a slower response time to odor changes in the water, whereas if a thin boundary layer forms around the rhinophore, the animal will have a faster response time to odor changes in the water. So one aspect that affects boundary layer thickness is the speed of water flow. And this pertains to my project because I was looking at how boundary layer thickness is formed at different speeds. So if a slow water current will allow for the building up of the boundary layer around the rhinophore, whereas faster water currents will tear off the boundary layer around the rhinophore due to turbulence. Tritonia are believed to have two behaviors that can manipulate boundary layer thickness. And the first one is what's called head sweeping, which is where the animal will wave its head side to side. And it's believed that by doing this, the animal will tear off the boundary layer. And also, there is cilia located on the rhinophores, and we believe that these cilia, we know that the cilia are able to generate its own current, and we also believe that that is also a way for it to tear off the boundary layer as well. Okay, so although we know that the boundary layer thickness can dictate how fast these animals respond to odor changes in the water, we don't know how often these animals are updating their sensory information. Due to the patchy nature of the odor plume causing a pattern of sit that comes and goes, these animals possibly have evolved in a response for the animal to have evolved a delayed response and averages their sensory input over a period of time rather than immediately when they receive the signal. Therefore, so the purpose of my project was to look at how water flow interacts with the rhinophores and Tritonia to gain an understanding of how olfactory cues that guide navigation are detected. So I had two objectives for my project. The first objective was to measure boundary layer thickness at various speeds. And we had hypothesis that with increasing speed, boundary layer becomes thinner. And we also wanted to characterize water flow turbulence. So how we planned on attacking this project was using a technique called particle image velocimetry, which I'll call PIV for short. And the basic concept behind PIV is measuring velocity fields and then measuring velocity fields by taking paired images that were taken shortly after each other and then calculating the distance individual particles moved around that time. So what I did was I took a model Tritonia and placed it in a flume tank that circulated flow in a mostly laminar flow. This technique involved a laser that emitted a very thin laser sheet that illuminated the rhinophores and the particles moving around the rhinophores. And attached to the flume tank was a video recorder that recorded recorded 30 frames per second as the water moved around the rhinophores. And so this is just what a typical video would look like. So you can't see it, but the laser was positioned here and it's illuminating the top of the rhinophore. And as you can see, along the sides here, the flow is mostly laminar. However, we do see some evidence of deflection around the rhinophore. And as you look behind the rhinophore, you can see that there is some turbulence. Okay, so here I have that same video that was uploaded into a software called Image J that just basically tracks the particles in the flow of water around the rhinophore. So here's the rhinophore here, and here we have a legend bar. So that describes the flow of water. So the blue and purple symbolizes low water flow speed, whereas the orange and the red signify fast water speeds, and the length of the arrow increases as water speed increases. So as I mentioned in the previous slide, again, this along the sides is mostly laminar flow, but as water flow is approaching the rhinophore, we see that there is a decrease in speed and also deflection around the rhinophore. So we see mostly red and orange arrows, which symbolizes fast water speeds, but as it gets closer to the rhinophore, it decreases in speed, which signifies that there's a boundary layer forming. And I measured the boundary layer, and it's about 2 to 3 millimeters thick. And also, we see evidence of turbulence behind the rhinophore as well. I didn't have, okay, so, sorry. Okay, sorry. So, the rest of my data was not analyzed, so we still need to analyze the rest of my data that I have. And for future, we need to analyze the rest of the flow patterns that I have in my PIV results to visualize other PIV results. Sorry. And we also, for future experimentation, we wanted to use a microscope in order to get higher resolution and better detail around the rhinophore. Sorry about that, you guys. Okay, so moving on to the second half of my project. So this man's name is Russell Wyeth, and he was the first to observe that the cilia located on Tritonia rhinophore were able to generate their own currents. So here I have some SEM photographs of some Tritonia tetraquetra rhinophores. So this whole structure here is the rhinophore. However, this feathery structure here is known as the clavus. And so this is the anterior part of the clavus, and this is the posterior part of the clavus. And we know it's the posterior part of the clavus because of the spire here. And as you can see in the close-up version of the photograph, that there's a little fine hair-like cilia So there's two types of cilia. There's sensory cilia, which is able to sense chemicals in the water, and then there are multicilia, which are able to drag currents along the skin of the rhinophore. So for this project, for this summer, I took up the challenge of characterizing the nature of the characterizing the cilia-generated water flow by using blue dye, and I did this on 3 species of nudibranch. So I did it on Tritonia tetraquetra, Tritonia festiva, and Armina californica. And so results generally showed that dye flowing from the distal end of the clavus to the proximal end of the clavus. More specifically, cilia was drawn in the dye into the interior region of the clavus, and then exiting out these vertical folds you can see here along the clavus. And just to know that this was done in non-flowing water. Also, okay, so here is an SEM photograph of Tritonia tetraquetra bear. I'm so sorry. So here we have a photograph of Tritonia tetraquetra's clavus. It's the interior folds of the clavus, and you can see here it's coated in what we believe to be multicilia. And you can see that the close-up of these folds is what dragging the current downward. Okay, again, so I also did dye work on Armina californica, and again, the general flow pattern was from the dye was flowing from distal to proximal. But however, unlike Tritonia, which would suck in the dye into the interior part of the clavus, the dye was sucked in onto the surface area, the surface part of the rhinophore. So this general flow pattern of dye flowing from distal to proximal is really interesting because when these animals project their rhinophores into the water column, it's possible that these animals are able to pull in odors from outside the boundary layer. So if this is true, that means that the animal can, this will help the animal respond faster to odor changes in the water. So in conclusion, there was a 2 to 3 millimeter boundary layer forming around the rhinophore. There was also turbulence seen behind the rhinophore. Tritonia has a more complicated rhinophore structure as seen than the other gastropods that were used in this experiment. There was also cilia on the rhinophore that was pulling from distal to proximal, bringing odor from outside the boundary layer. And for future work, I would like to characterize cilia-generated flow in other aquatic gastropods, as well as characterize the difference between motile and sensory cilia. And by doing this, I would hope to better understand the role cilia has in odor-based navigation. And we also wanted to use a microscope for higher resolution and PIV projects around the rhinophore so we can get greater detail. And I would like to thank all these people. Any questions?
We have a couple minutes for questions. Any questions?
Yes.
So how is this gonna help? What's the difference between the Armina and the Tritonia as far as how they like to live and crawl around?
What do you mean?
I mean, what about how do you think that would work with burrowing with the Armina?
Okay, well, so Armina burrow, so if they stick their rhinophores outside of the sand, they can get odors that way, whereas, with Tritonia, they don't really burrow, so they can just use the odors from outside the boundary layer, I guess, if that makes sense.
One more question.
I was just, I think I'm asking a similar sort of question, which is what's the types of odors that the Armina would be smelling versus the Tritonia? In other words, I assume it's mostly prey, but do the prey odors theoretically come in differently for the two of them that would dry it down?
I don't think that they would. I'm not very sure, but I don't think that they would.
What does Armina feed on?
They feed on chickens.
Oh, they both feed on.
Yeah, they both.
Oh, okay, so yeah, then prey.
All right, thanks a lot. All right, our next presenter is Will Schneider from California State University, East Bay. His mentor is also Jim Murray.
So I spent this summer studying the possibility of magnetoreception in the nudibranch Armina californica. So what is magnetic sense? Magnetoreception is the ability for an organism to detect the magnetic field of Earth, use it to orient itself for the purposes of navigation or just spatial positioning. There are creatures like the roundworm C. elegans that uses it to optimize the direction that it digs its burrow to maximize depth. There's animals that use it to migrate great distances in the ocean, like the large sea turtle and the smiley lobster. So a big question these days, and for a long time, is how is this possible? And not exactly surprisingly, the answer is magnets, or very, very small magnets. Small crystals of magnetite, which is an iron oxide compound that's known to be produced by biological organisms. This is a cell from the nasal passage of a rainbow trout. At the end, there's a little pocket full of magnetite, which is a magnetic substance. And this is a neuron at the end of C. elegans. This is kind of exciting stuff. This is one of the few known primary magnetic receptors, meaning that this would be the structure that is accepting magnetic stimulus. And of course, everybody knows and loves Tritonia. Tritonia is also an organism that can sense the magnetic field. And as Teris has already demonstrated, this animal is fantastic for neurobiologists for a lot of reasons. But mainly it's because it has a small brain of around 7,000 neurons, but it displays a rather wide range of sophisticated behaviors, including escape swimming, flow orientation, and geomagnetic orientation. So how do we know that they orient magnets? In 1987, Lohmann and Willows demonstrated that Tritonia, when disturbed or dislocated, in the absence of stimuli, would group together, they would be facing in direction as a group. And this was a significant result. And then when the geomagnetic field was canceled out by a coil in the opposite direction, the response was abolished, and they appeared to distribute themselves randomly. So they didn't stop there. They went on to find the cells that were associated with magnetic receptivity, and they discovered that these cells on the left and right side on the pedal ganglia, PO5 and PO6, are excited by changes in Earth strength magnetic fields. It turns out that these cells are also associated with crawling among gastropods. They're part of a motor network that modulate the beat frequency of cilia on the foot of the animal. So the animal moves along by letting mucus and then using cilia to push through it. And these networks over here are conserved among most nudibranchs and even some non-nudibranch gastropods. So why Armina? Armina and Tritonia are both nudibranchs in the subset known as cladobranchs. They're found in the same habitat, same soft sediment and muddy bottoms associated with sea pen forests. They both eat the same sea pen, Tylosarcus gurnii. This is down in the lab, had a bit of a feeding frenzy going. And they both use mucus and cilia to move along on the bottom, just like most gastropods do. So remember that I said that that motor network is conserved, and this is true across many, many organisms, or gastropods anyway. So Most of the nudibranchs exhibit this conserved network that we've tested, and even some non-marine terrestrial gastropods, the Stylomatophora, also exhibit this conserved network. So more so than that, in Armina and Tritonia, this network is actually homologous, so meaning they have the same function, they have the same location and structure, and they appear to be very, very similar. So these two cells, which are excited by magnetic stimuli in Tritonia, are known to be homologous with these two cells in Armina. This is a whole brain of Armina. So on the left and right side are the pial ganglia, and then this is the left pial ganglia here and the right over here, with a close-up on the pial PPD1 cell on either side. You may be able to make that as this slight pale circle here. And then on the right, this is that PPD cell with an electrode inside of it taking an active measurement. So how would we go about testing to see if these animals are receptive to magnetic stimuli? I started with the physiology. So I did what was called a semi-intact animal preparation, where I exposed the brain of the animal with an incision and immobilized it in a dissection tank that had the magnetic coil around it so that I could rotate it when I was ready. I had to take great care not to damage any of the nerves because the cells, if you remember, are correlates of activity. The cells themselves are not responsive to magnetic stimuli. They're receiving stimuli from somewhere else. So if I damage any of the nerves, I would likely have to start over or I might not see any response at all. So when I had the brain exposed and cleared, I had it pinned onto this non-magnetic wax platform, and I used a glass microelectrode to get inside of the cell so I could directly capture readings of the behavior. So these are action potentials from the right PPD cell of an Armina. So this is about a scale of about 5 minutes. And the idea here is that if I rotate a magnetic field around the animal while I'm recording, and they are receptive and these cells are truly homologous, we would see an increase in activity or spike rate of the animals. So I'm looking for an increase in spike rate associated with magnetic stimuli. Each of the trials is 30 minutes long. I start with 10 minutes of ambient recording period, followed by 10 minutes of active rotations with 1 minute per rotation, or a control, and then 10 minutes of ambience again to compare to. And for each neuron that I test, I need at least 2 trials of control and an active rotation to make the comparison. So in the end, I had 8 total neurons tested, and each one of these preparations might take upwards of 3 hours, and I have to record for at least 1 hour from each one. So this was quite a feat for me. I wound up with a total of 24 trials, but only 8 individuals, so we had to do non-parametric testing. And what I observed in the control was that the cells themselves would slowly decrease in activity. They were still healthy, but there was generally a decrease in the rate at which they would be firing over time, and after about an hour, they would usually go silent. And there was no increased activity during the control rotation. Not a big surprise. The control rotation was activating the field in the direction, the same magnitude as the natural magnetic field, which we mapped. So no big surprise there. And the rotation controls, the same thing happened. There was a similar trend of decreasing activity. Eventually the cells would go silent, and there was no significant increase in activity associated with the magnetic rotations. But I wasn't convinced. I wanted to be a little more thorough, so we went back and recreated Lowen's experiment here, where I disoriented some sea slows. Put them into a flume tank and swirled them around in a sort of haphazard fashion to try to arrange them as randomly as possible. And then we recorded them in time-lapse using video software afterwards to note their positions at 10 minutes, 2 hours, and 4 hours after the disorientation to see if there was any significant grouping in their orientation at any of those points. In the original Loman and Willows paper, they checked only in 4 hours, but I thought it might be beneficial to look beyond that. In addition to this, I noticed that some of these animals come to a stop, and to me, they seemed like they were choosing these nice angles. So I decided to look a little harder at the directions that they had stopped in, periods at which they stopped, like why were they resting, what was going on. So for the timed areas, this is what the data looks like at 10 minutes, 20, or 2 hours and 4 hours. These are the actual angles of the animals. You notice that they're not very graceful. A lot of times they'll be on their back when I go to check their position, so we can't exactly use that for the data. So to simplify that a little bit, this is what their mean groupings of their directions look like. And to simplify it even more, none of them are significant. We got a little close here, but nothing that could truly convince me. So there was no significant grouping during the time experiments, so I thought, well, what about when they were stopping? Is there any grouping to how they stopped? So this is a graph that I made of the time spent resting, the angle at which they were rested during the control and the rotation trial for 10 individual slugs. So this allowed us to look, to compare before and after for the whole group and for individuals across time. So within the control and within the rotation, there was no grouping. And I went further and we wanted to look at, orientation across individual slugs, and within that we also found there was no significant orientation. So, what can we conclude? The evidence is not looking good for this case. It seems that Armina does not, if it does sense magnets, it shows no trace of it. And the reason why we wanted to compare it against Tritonia is because Tritonia is a wonderful model organism for studying all kinds of neurobiology. And it would be kind of interesting to see if it is really unique in its ability to sense magnetic fields. And that's one of the questions that I had looking forward. But another one is, why would a nudibranch species be able to sense a magnetic field? What kind of adaptive significance would that have? These animals don't migrate enormous distances. To my knowledge, they don't burrow very deep. So why would an animal like this have this behavior? And is Tritonia the only one? Are there others out there? We haven't done too much surveying of this sort, but it would be interesting to know if they are truly unique in that regard. And how would a sense like this arise in an animal? What kind of structure would lead to the ability to sense magnetic fields? What kind of significance would that structure have on an animal? And are these senses conserved from previous past, or are they derived eventually? Especially in regards to Tritonia, I'm curious about that. Yes, and I would like to thank all of these people, including Jane Murray. It's great. Teris, thank you so much. And all the directors and participants of the REU program. You guys have been great. Questions?
You showed the classic study with the orientation of is that been replicated? Are you convinced of that result?
I have not myself performed the replication, but yeah, it's been done many times.
Okay.
And it actually turns out that the animals would be oriented in a direction, but the direction would be different most times that they perform the experiment. And in the same paper that I took the figure from, they go on to conclude that it's correlated with the lunar cycle. So the animals might be grouped and headed in a different direction based on the cycle of the moon. And that was a paper published in 1987. And there's subsequent follow-ups with the arrow stuff too.
So I think there's a question
But
in
the
the back.
behavior is all about, so the behavior.
Yeah, I mean, it makes one wonder.
The back, yeah.
Yeah, that was my question. Are you against original work? Magnetic perception was related to the lunar phase, and when did you do your work?
I did I'm sorry, what?
Did you look at the moon?
No, that would have been a very prudent thing to look at. I did all of my experiments within 1 week of each other, and I did them at the same time of day, from 9 p.m. to about 5 p.m. But that was as much as I controlled for. I also noticed that there was no obvious grouping in any situation, regardless of that. So it seemed like they were orienting kind of randomly, depending on how the field was.
All right, thanks a lot. All right, our next speaker is Noah Bressman from Cornell University, and his mentor is Alice Gibb.
Come on.
So my project is a comparative analysis of the novel terrestrial locomotion of the type of sculpin, Oligocottus maculosus. So why study fish out of water? The intertidal life poses many challenges for fish, such as hypoxia, desiccation, rapid temperature changes, and salinity changes. So by studying their adaptations, we can study how they can survive in this harsh environment. Well, studying fish on land also is a great analog for studying how life first transitioned from water to land. So here's the typical sculpin, cute little guy right there. They live in tide pools high up in the intertidal zone. They appear to use actual appendage-based terrestrial locomotion, meaning they use their appendages and their body in terrestrial locomotion, but no one has described this yet. So here they are in the field. You can see they are squiggling around with a goal in mind. They're moving in a specific direction, but it's hard to tell how they are moving. They seem to just be squiggling around in these tide pools. So how do they move? We use high-speed video to analyze their kinematics, which is their motion, to describe their form of terrestrial locomotion. We also use landmark tracking in MATLAB to track points along the body. Using the tracking software, we measured several characteristics, such as the wave amplitude as a percent of body length, which is the amplitude of the wave at different points along the body as a percentage of the total body length. The distance ratios, which is the net distance traveled over the total distance traveled, to show as a measure of how linearly they can move. We also measure the stride frequency and the curvature coefficient. The curvature coefficient is the bend length from the snout to the caudal peduncle, the base of the tail, over the straight length. And we also measured velocity. So here is a type of sculpin. This is slowed down 7 times. You can see its terrestrial locomotion. So they clearly are moving with a defined characteristics, which are they will rotate their body around a planted pectoral fin, and then they will drive with their tail. I'll show you that again. They rotate with the tail and pectoral fins on the same side using a lateral gait. They also will lift their body off of the substrate using ventral flexion and by rolling slightly along their long axis onto their side so they can push their caudal peduncle against the substrate. As you can see here. They are lifting their body clearly above the substrate. So you can see that they move by alternating pectoral fins. They rotate about the pectoral fin. You can see the pectoral fin stays stationary, whereas the body rotates around the pectoral fin. And it's all driven by lateral oscillations of the tail. They use a lateral gait, the pectoral fin on the same side as the tail, as making footfall. In this instance, the tail acts as 2 legs, one leg on one side with boost on one side, and another leg has boost on the other side. The only other organism, the most common other organisms that use this type of lateral gait are giraffes, camels, and elephants. No other fish has not been seen in any other fish. They also use ventral flexion, which allows them to lift by tightening their belly, they can lift their body off the ground, and then they can turn and push off the caudal peduncle in order to move forward. They employ a standing weight, which you can see that alternates around the body as the same. And we want to see how does their terrestrial locomotion compare to their aquatic locomotion. So here's a quick video of how it moves in the water. Initially, with its aquatic fast start, it rotates around its pectoral fin and moves its tail to the same side. which seems to look like once a half stride of the army crawl, which is what the Oligocottus. And so it appears that the origin of their army crawl originates in a modified series of aquatic fast starts adapted for terrestrial locomotion. However, during steady swimming, they use lateral undulations and keep the pectoral fins to their body and waves along their body to move. So you can see they use a standing wave on land, where the wave amplitudes are greatest at the head and tail, and in water the wave amplitude increases as the wave travels down the body. We also want to see, why do these sculpins move better than subtitle sculpin species? So this is a Pacific staghorn sculpin, which sometimes can be found, the young ones can be found in tide pools, but it generally lives in the shallow subtidal zone. And this is the northern sculpin, which appears to have a similar overall body shape to the typal sculpin, yet it lives very subtidal. So here's a video of the staghorn sculpin. Their terrestrial behavior involves lateral oscillations of the tail and head, which we call a wiggle. The wiggle, as you see, achieves little to no displacement. It simply wiggles around the center mass in place. The northern sculpin will sometimes do the wiggle like the Pacific staghorn sculpin, but sometimes they'll do what we call a tail jam wiggle, where as their tail approaches maximum amplitude, they will rapidly accelerate their tail towards their head in a jamming motion. So there are some differences between the pectoral fins usage and morphology between these species. The typal sculpins have a smaller insert than the staghorn sculpins, which allows them to have more freely rotate about their pectoral fin. They also have adaptations for grip. They have reduced ventral webbing compared to the staghorn sculpin, which allows them to get a better grip on the substrate when they're rotating. While there is no, these figures are from a paper by Kane and Gibb, but they did not do any, there's no information on the pectoral fins of the northern sculpin. However, the northern sculpins do not move their fins, and neither do the Oligocottus and staghorn sculpins when they are performing terrestrial behaviors. And the typal sculpins move much more quickly on land. As you can see, they move many more body lengths per second, and they move up to 10 centimeters per second, whereas the other two species move about 1 centimeter per second. So they're much more efficient at moving quickly over land in a linear direction. They also bend the typal sculpins also bend their bodies less than the staghorn northern sculpin, so they lose less energy laterally and focus it more interiorly. So how do these fish compare to other amphibious fish? So this is a walking catfish. There's been a lot of research done on the walking catfish terrestrial locomotion. And here you can see they do another form of actual appendage-based locomotion, where they rotate around they use a diagonal gait and rotate around their outside pectoral fin and bring their tail. So they use an opposite gait as the typal sculpin. However, there are some similarities. Their tails have similar wave amplitude, and they also have a similar distance ratio. So the tails are moving same amount laterally as they are anteriorly for both species, for all three species. However, the walking catfish have a much lower wave amplitude and a higher distance ratio for the center mass and head. This could be because the walking catfish rotate around the tip of their pectoral fin, whereas the typal sculpins rotate around the base of their pectoral fins. So by increasing the radius of their pivot point to the center of mass, the walking catfish can rotate the same number of radians, but cover a larger distance than the typal sculpin, which could account for the differences in wave amplitude and distance ratios. To summarize, the novel mode of terrestrial locomotion by the typal sculpins is characterized by a lateral gait with rotation about the pectoral fins, driven by lateral oscillations of the tail. Their terrestrial locomotion is different than their aquatic locomotion. In an aquatic environment, undulation works well in a fluid environment. However, on land, they need to overcome gravity flexion and rolling onto the side and pushing with their caudal peduncle. That allows them to lift their body up and overcome the friction. They also, the origin of their army crawl likely relies in their aquatic fast starts. The sculpins that occupy the higher intertidal zone, the tidal sculpin, are much more effective at navigating and moving in their terrestrial environment, which allows them to return to water much more quickly or avoid predation when on land than the other species. So this study provides behavioral evidence that terrestriality could have preceded terrestrial morphology. Early fishes could have moved onto land prior to species like Tiktaalik before the rise of modified forelimbs just by using behavioral modifications. In the future, we would like to do a more in-depth analysis of the human leopard crawl. We want to do this because the human leopard crawl appears to be similar to the tidal sculpin's army crawl, which is why we call it an army crawl. However, tidal sculpins, they're very small, so it's hard to look at the ground reaction forces and the biomechanics of their terrestrial locomotion. So by studying humans as an analog, we'll be able to understand better about how they move on land. We'd also like to use modeling, such as we started with this, what we call the cedar sculpin, as a simple wooden model to show the terrestrial locomotion, how they rotate out their pectoral fins. Currently, we are developing a Oligocottus, a robotic sculpin that will move in the same way as the Oligocottus type of sculpin. We also did some other research describing the natural history of other species of sculpins and describing their terrestrial locomotions. Here is a silver-spotted sculpin performing a tail flip jump, where standard tail flip jump where it is a modified sea start on land. And here is a new locomotive behavior, which is called somersaulting. This is by the Dasycottus sculpin, where it will roll over its head, like so, and we will continue doing that and moving downhill. Any questions?
So why did you choose to compare tidal sculpins to like deeper water species?
We wanted to see if deeper water species, even the ones that look similar, will perform the same behaviors. We wanted to see if they have the capability of moving on land effectively. But we needed a comparison for the sculpins. If the subtidal sculpins move with that same behavior, then it could be a trait of all sculpins, but as we found, they do not.
For your deeper water sculpins, are they swimming underwater the same way that they're attempting ineffectually to walk on land? But the tidal sculpin switched both ways, switch what he's doing. Do the deeper water ones also try to switch?
We haven't looked into that, but it appears that the sculpins generally use transverse waves within the body, undulation, and whereas on land they are also exhibiting a standing wave, like the tidal sculpins. They oscillate the body back and forth rather than undulate. So it seems that they're doing different things than in water.
All right, thanks a lot. All right, our next speaker. Our next speaker is J.D. Laurence-Chasen of Brown University, who was working this summer with Beth Brainerd.
So I had the pleasure this summer to work with some pretty cool fishes and some pretty cool people. So I'm going to jump right into it. There are 30,000 species of grayfin fishes. And for some context, that's half of all living vertebrates. That's a lot. And despite some pretty incredible differences in cranial morphologies, I mean, you can see the trumpet fish and the slingshot grass, which is totally different heads. Despite that, the vast majority of those 30,000 species all exhibit employ the same behavior of prey capture, same method of prey capture, suction feeding. So as a functional morphologist, two questions come to mind. First, how does it work? How does suction feeding work? And second, how does the head morphology, the cranial morphology, how does that inform, how does that impact suction? So I looked at sculpins, similar to what Noah was just looking at, and they have, kind of squat, like flat heads, kind of little like toads, very big mouths. This is what Oligocottus looks like. Ooh, yeah. What a stripe looks like. In slow motion, shot at 250 frames a second. Play it once more. So you can see the rapid expansion of the mouth and depression of the jaw. So that's what I'm focusing on, the depression of the jaw. And in case you're curious, what it looks like in real time, it's a bit faster. It's pretty cool. There's a reason we need high-speed video. So kind of the main players, what I'm going to be talking about first is the operculum. So that's just this kind of triangular bone. It sits right on top, it covers the gills. So if you ever see a fish doing something like this, that's the operculum that's moving. And then the suspensorium. So the suspensorium suspends, makes sense, suspends the lower jaw from the cranium, the neurocranium, as it's called in fish. There's a few more bones. There's a lot more bones. So the red circles here are muscles, the green squares are bones, and then these three kind of bluish squares are the actions that are a result of these bones and muscles working together. So the bones and muscles in a fish's head, all have their own kind of unique 3D motions during a suction feeding strike, and they work together in what we call musculoskeletal linkages. So I'm going to be talking about one specific linkage, the opercular linkage. And what that involves is muscles generating force way up in the back of the head, and that force being transmitted through several components in the head, resulting all the way across the front of the head, resulting in jaw depression. So this theme of force across a distance is pretty prevalent in suction feeding in the teleost head, and so it's really cool. We want to learn more about it. We kind of honed in our questions now, right? So it went from how does suction feeding work to how does the opercular linkage work? And instead of what's the relationship between form and function, what's the specific relationship between cranial anatomy and this specific linkage? Another image that demonstrates it, I think, a bit more clearly is this. You can see the operculum right here. You can imagine how it might rotate up right on that opercular hyomandibular joint. You can see how it would rotate up, and that would pull right here on the interoperculum, put the interopercular mandibular ligament in tension. Let's say that 5 times fast. Put that in tension, and that pulls back on the lower jaw. The lower jaw would then rotate down around that quadratomandibular joint. So this linkage has been modeled as a 4-bar linkage. A 4-bar linkage is a specific type of mechanical model that's planar. It describes this motion relatively well at first glance. So we can see that this green bar right here would be the operculum rotating dorsally, and then the coupler link, which is this red bar right here, is going across here, and that pulls on the lower jaw. And this distance, this little red bar, or this little yellow bar right there, that would be the distance between the quadratomandibular joint and the interopercular mandibular ligament. So that is that little bar. You can imagine how as it pulled, the whole lower jaw would come down. So this is a very robust mechanism. So robust that if you were to pull up on the operculum of a dead fish, the jaw would depress quite significantly. I did this in a series of manipulation studies. You can see a string attached right to the tip of the operculum there. You can see these beads mark the operculum. And there's a screw in the head into the neurocranium postmortem. This is a dead Homologous to stabilize it as I activate this opercular linkage. And so we can look here, see quite dramatic jaw depression. You can really see kind of just along here, that link is really moving. The two species that I looked at specifically were Homologous, which you just saw, the red Irish lord, and the Pacific staghorn sculpin, or Leptococcus armatus. Every time you hear it, just Leptococcus armatus. They're both beautiful fish, but with very different cranial morphologies. So looking at a CT now, so 3D models of their cranial bones, you see a lot of differences, but one I'd like to highlight is going back to that little yellow bar, that output link of the 4-bar linkage. You can see, looking at Homologous, there's a little kind of, there's a nudge, a knob that comes out there. We call that the retroarticular process, back of the articular bone, which that is. process coming out, which is not present in the Oligocottus. So what that's functionally doing is lengthening this output. So that's a main difference in morphology. Another thing that I'd like to highlight that kind of jumps out is the fact that the four-bar linkage, as I mentioned, is a planar linkage. But these bars, as you would draw them in this fish on the skull, are three-dimensional. They're not planar. A fish's skull is very three-dimensional. So we kind of run into this problem we're trying to understand how this works, how this linkage works. But if the model is planar, we're definitely going to lose things along the way. So how do we evaluate what's going on in a fish's head in 3 dimensions? We really wanted to answer that question, so we spent the summer working on a new method that we call VROM, or Video Reconstruction of Moving Morphology. And this is a way in which we can generate precise animated 3D models of these complex 3-dimensional motions. This is based on XROM, X-ray reconstruction of moving morphology, a technique that was created and developed by Beth Brainerd at Brown. And what XROM involves is biplanar X-ray videos, which result in the creation of these highly precise 3D animations. So this is a largemouth bass strike that Ariel Camp and Beth Brown worked on. It's beautiful. So how does VROM work? Well, what we did here, we started with marker attachment. So we attached markers to each of the bones of interest. We attached 3 markers to each bone so that we can get that bone's motion, capture that bone's motion. So we can see here the operculum, 3 markers right on there. And you can imagine that as the operculum is moving, those markers are moving along with it. Second step, data recording. We take high-speed videos, 3 individual cameras at 500 frames a second. And actually, it's a cool thing, a few seconds after I took this picture, we see this. It's really amazing. You see how the prey is going, the piece of shrimp it is, going right into the mouth. From 3 different camera angles, you can see how the markers are each visible in a different view. Right on. So after data recording, we get into some tricky stuff, calibration. So we spent a lot of time and we are still working on this because it turns out that the refractive nature of water makes shooting video through that in 3D camera positions quite difficult. We're currently trying 3 different calibration objects. Thank you, Adam Summers, for constructing this beautiful lattice object that we're working on. We have this Lego object. Thank you, childhood, for that. That's really great. And then we're using a checkerboard as well. And then all this testing could not be possible without our main man, Dr. Ben Norland, over here to help us with that. He's brilliant. He is, as he likes to remind us, I'm not a biologist, but he still loves fish. And so Ben created this software that we use called XMA Lab, which is where we do the marker tracking. So this is a screenshot of what that might look like. On the left here, we have all the different points that I'm tracking. You can see the green kind of crosshairs on them. And then you can see these red lines connecting the points that are a part of the same bone. So we call those rigid bodies. And what the data that we get out of this program are what we call rigid body transformations. So rather than the 3D location of a single point moving, it's the 3D translation of the rigid body of the bone itself. And then an ultimate step is a CT scan from which we create 3D models of the bones. Finally, we take those rigid body transformations, so what we got by tracking the markers, we take those and then we apply them to the 3D models we got from the CT scan. And the result is a pretty beautiful looking 3D animation from which we can measure the rotations and translations of specific bones. So what did I do? Well, I first compared a live Leptococcus to the manipulated Leptococcus. And my prediction was that the live Leptococcus would have a greater KT than Leptococcus am. So KT is kinetic transmission. And what that simply means is the ratio of input rotation to output rotation. So when we were looking at that 4-bar linkage, we saw the green bar going up and the yellow bar going down. Green bar going up is input rotation. So a high KT, high ratio would mean that for a given input rotation, there's going to be a lot of jaw depression. There's going to be a lot more output So what did we find? So this is the live leptococcus data, and we saw that that was definitely the case, that there was a very high ratio. Kind of just dissecting this graph a little bit, we see that there are two lines, two curves going on. Each curve is a different bone, and its rotation about a single axis through time. So this smaller curve is the operculum, rotating dorsally, and then the larger curve is the lower jaw, then depressing. And so you can see, looking at this graph, that the operculum does, a bit of rotation, and boom, jaw goes down. We say that KT is 4, so 4 times as much lower jaw rotation. Let's see, so now comparing this to the manipulated leptococcus, we see that the KT for the manipulated leptococcus is 2.3, significantly less than what we observed in the live leptococcus. What that actually means is, as I pulled up on the operculum, the lower jaw went down twice as much rather than 4 times as much what's happening in linkage. Second set of predictions, comparing Homologous, the other species, with leptococcus. So if you remember that retroarticular process, here I mentioned kind of there's that little nub, so I hypothesized that that nub would extend the output link. In a longer output link means that there isn't going to be as much rotation. So I said that the leptococcus with a shorter output link would show more rotation or a greater KT ratio. And that is what we observed. Looking at Homologous up here with the KT of 2.0 and leptococcus with KT of 2.3, 2.3 being larger. So in the leptococcus, with a shorter retroarticular process, there was more lower jaw depression for the given opercular rotation upwards. So the predictions, success, confirmed, always good. The two manipulated species were similar, but as I predicted, the leptococcus had a larger KT value. And so the live leptococcus also, as I predicted, had a significantly larger KT value. What that means is there's other things going on. There are other mechanisms going on to power that lower jaw depression, that lower jaw rotation. As you can see from this picture, there's a lot going on in 3 dimensions. It's not just kind of a planar down. And so suspensorium, lateral expansion, that can be a cause, and then also the hyoid can be a cause of that. And something that also suggests that is when we go back to this first graph of these rotations, you see that the peak of the opercular rotation is well before the peak of the lower jaw depression, the lower jaw rotation, which suggests that, something needs to be happening right here that makes this go down further. Further studies. So these are the predictions, but also super important. Neuron works. This was huge. We spent the summer working on this, and it's really big. The fact that we can, without $500,000 X-rays, create really extremely precise 3D models with, as Nick so eloquently put, with iPhone cameras. We really tried really hard. Which is amazing. And then future directions, there are a few possibilities. So thank you, and thank you, Beth Brainerd, my fabulous mentor, Yordano, my partner in crime, and everyone else who had our lives. Yeah.
Yeah, we have time for a couple quick questions.
As we spent a lot of time figuring out the method, we only have time for a lot of trials because it's a process of marker attachment and all that stuff. So the dream is to do the live videos and the live 21,000 hours. Yes.
Yeah, I remember Carl Lee back in the 1980s talking about 2 or 3 or 4 different musculoskeletal circuits by which fish milk in their mouth.
Yes.
Can you comment on the applicability of this method to analyzing different sets of muscles and bones?
Yeah. The opercular linkage is probably the most applicable, the easiest, just because of the reason that we can do VR, which I should have mentioned in the thing, is the fact that the fish's bones are very superficial. That there's very little, that we can attach markers, and that those marker, movements are essentially the bone movements. So when we're talking about the hyoid linkage, you're not really going to put a marker on the external surface that's going to kind of act. I guess kind of eventually you can see something, but not quite as well as the appropriate fish.
All right, thanks a lot. Next talk is Yordano Jimenez of Northern Arizona University, although soon to be a PhD student, starting this fall at Brown University with Beth Brainerd.
So I'm Yordano, and I spent this summer working with Dr. Beth Brainerd looking at how the vertebral column of fishes bend when they suction feed. So suction feeding is simply put just a common form of aquatic prey capture, and it is employed by various organisms such as sharks, turtles, amphibians, and fishes. In fact, most fish are suction feeders. And what characterizes suction feeding is the creation of a negative pressure inside the mouth by rapidly expanding the jaws outward. And so you can see this rapid jaw expansion and the fish getting sucked right in. And I'd like to focus on one component that contributes to this suction feeding, and that's the head elevation that happens. So head elevation takes place by first the contraction of these epaxial muscles on the sort of upper body of the fish. And when these muscles contract, they yank on the neurocranium, they pull it up. As a consequence, this, through the linkage that I'm not considering at this point directly, but through some linkage here, the hyoid swings open. And so all of this works through the contraction of these epaxial muscles. And I should note that these epaxial muscles are no small matter. In fact, they are contributing significantly to the production and generation of this suction during feeding. In fact, the paper that came out by Camp and Brainerd showed that it's, in some cases, could be up to 90% of the power required to generate this suction. It's quite a large amount. And so this is what the anatomy looks like when all of this is happening. And there's a lot going on. I just want to talk about the neurocranium and the vertebral column. And it's quite difficult to sort of just analyze in the full complexity what suction feeding is and what bones are moving, which muscles are contracting, which joints are moving and just how much. But just these two are going to be the focus of my talk. And to show what this would look like as if we had X-ray vision, you could see all these moving bones. So to find how much the vertebral column is bending during suction feeding, we first should find out and figure out where the axis of rotation is during head elevation. So here's a trumpet fish lifting its head up during suction feeding. And the question is, well, if the axis of rotation were here, then we would know that all the bending is sort of happening between the head and the body. However, if the vertebral column is bending, then the axis of rotation moves back because that part is also bending. And we sort of wanted to see how this applies to the diverse morphology we see in fishes. Not just cranial morphology, but vertebral and axial morphology. In some cases, you can see just how robust these elements can be here. And so you would wonder, how is that going to rotate? How is that going to allow for any head elevation? And in some cases, you can see, okay, I see a little bit of wiggle room there, and some, you're not quite sure. But we wanted to look at how these structures affect head elevation and bending of the vertebral column. So to sort of little exercise in identifying the axis of rotation. Here's a video, and let's focus on the body plane and the neurocranial. And as this video plays, try to identify the axis of rotation. I mean, yeah, you know, I could say somewhere in this vicinity, but other than that, there's not much else to say. We have to actually analyze this in more depth. So, in comes using joint coordinate systems. So, we acquire data using VROM, and once we have our animations, as we've been seeing throughout, we plot these joint coordinate systems to the animations. And what these joint coordinate systems do is they tell you how this part is moving relative to this. And so we plotted a bunch of points where the axis of rotation might be, and we singled out just
So on the laser pointer. Yeah, yeah, there you go.
We isolated just that one point that worked the best. And so how do we know which point is the axis of rotation? Well, when you run these joint coordinate systems, they tell you how much it's translating and rotating. And so we wanted to find the point which was translating the least, because that would be the axis of rotation, and the one that was giving us the most rotation, which is the one that's actually representing the head elevation we see during the strike. So here's a video sort of showing what this looks like. There's here. And so this pink axis is actually the, this would be, it's showing you how much it's translating. And this other one remains fixed in its position, this blue, green one. And so this is a pretty solid axis of rotation. There's very little translation compared to the rest of them. So we repeated this for 3 different species of fish. And we did the largemouth bass, the staghorn sculpin, and the striped surfer. Right off the bat, we start to see that there are differences in the vertebral morphology. And so we ran through and plotted the different joint coordinate systems for each of these fish and identified axes of rotation. So just to sort of orient ourselves, we're going to look at the largemouth bass. And these are the first 4 vertebrae. If I'm looking at this picture and I had to guess where it's going to be rotating, where would it be? Where would that axis of rotation be? To be honest, I mean, past this point, I just don't see that much wiggle room. I mean, the neural spines, the pterygophores, these other spines coming from up above, they're sort of interdigitating. And so you don't have that much space to be bending in that location. However, I do see plenty of room here for bending. And if I even had to be more specific, I would eliminate this joint right here between this first and second vertebrae. And the reason for this is because, you know, they look sort of bulky there. You just don't see that much room, as opposed to at least this spacious area. So I would just guess, all right, you know what, I'll put my money on that. And, well, give me my money now. So that's where you find it. We had 3 different strikes, and we identified the axis of rotation for each of those strikes. And this is then being plotted on the 3D figure. And they are between the 2nd and the 4th vertebrae. And let's sort of recap where that is. Right here. All right, we got a staghorn sculpin. All right, this is looking a little different. Same thing here. First 4 vertebrae being pointed out. Right off the bat, I'm just going to say, I don't know, maybe past the 3rd vertebra, I'm not going to see that much rotation. That's not going to be our axis of rotation. And well, it's sort of constrained to between the first and the third vertebrae. And so it's sort of just, conveniently working out that the morphology is telling us where they're bending, which is great. That's exactly what we wanted. And now last we got the surf bridge. Same thing, first 4 vertebrae, and this looks different. We have all these neural spines popping out, pterygophores coming down. Where is this fish going to bend? And honestly, I'm just not entirely sure, but if you look closely, you see this little opening right here. Not so much right here. The space is sort of consistently narrow, but here there's a small opening. I don't know. I'm just guessing. That's where it's going to be. And, well, it's right around that area. So that's where you get the bending. That's where the axis of rotation is during suction feeding of these fishes. And so it's a pretty interesting story. It's fascinating how the morphology is sort of leading us to where these answers are. And VROM has supplied us with a sort of tool for analyzing this. And it's really, really cool. And so just imagine how this would apply to looking at this, at the 30,000 species of fish with fishes with different vertebral morphologies and different abilities to suction feed, and seeing how this can help us identify where they're bending and how that relates to morphology, which is what you would expect. And I'd like to tie this back to something very interesting. These epaxial muscles that cause the head to lift and the hyoid to depress, basically contributing to sort of the expansion of the jaws. These muscles are the same muscles that have been normally associated with swimming. And that's very, very interesting because now you don't just have epaxial muscles and the vertebral column. Now you also have, they're not just contributing merely to swimming, but they're also contributing to suction feeding. So you have this sort of multifunctional role of the vertebral column and the epaxial muscles, which raises many questions. Now you start asking, well, if this structure contributes to more efficient swimming, then how does that affect potentially feeding, or questions of that sort. So with that, I hope I got you thinking about vertebral bending during suction feeding, and I'd like to thank Beth Brainerd for sure. I mean, she's been awesome. It's been great. Gonna be my PhD advisor, so I'm really excited about that. That's really cool. JD, partner in crime, that's right. Ben, Norland, Vic, and Adam for sure, and just the whole REU program in general. I'm very grateful. And that all questions.
So this one comes from our live feed from the Twitterverse. Someone wants to know whether it's possible to get any finer resolution on those neural spines.
Like on the CT scans and such?
Yeah, is it possible to figure out more about the movement of those with these external markers? Is it?
I think it, let's see, with the external markers, not so much. So what we did is we had the animation of the body plane and of the neurocranial, and we found the axis of rotation in that space. And then what we did is we sort of imported the vertebral column into position, and that's where we sort of infer that that's the vertebra that's bending, or at that joint, I should say. So yes, external markers are not going to give you sort of a high resolution in that respect, but sort of perhaps rotoscoping or something of the sort could help clarify what the bending's happening.
Yeah, so the way you define the axis is where you maximize the rotation and minimize the translation, right?
That's correct.
So that's a geometrical reduction of what could be more complex movement, right?
Yes.
So you're not claiming that the bending is confined to that.
Absolutely not.
But
yeah.
That it's distributed over several vertebrae.
Right, yeah, I think the best way to describe it would be to say that the bending is sort of centered there, but not limited to that particular joint, right? And yeah, I'm sure these joints are bending.
All right, thanks a lot. All right, our next speaker is Sara Magallanes from California State University at Monterey Bay, and her mentor is Diane.
On predatory interactions of two predators on two species of lacuna on the Rocky Mountain community around the Catalan Islands. And so why is predation important? Well, for one thing, it helps prevent a single species from dominating an area. And a great example with this is with Magallanes, californius, and Tess Dooley. And so the removal of these mussels by these sea stars creates open space for other organisms to come and inhabit, and you get a control of the prey population. And overall, a higher biodiversity in the area. And so our selected prey species were two species of lacuna, lacuna lynx and lacuna variegata. And a great way to distinguish the two is that for variegata, you see these chevron patterns on the shell. And they typically don't get any larger than 9 millimeters in length, and that's quite small to work with. So. And they both live in eelgrass and micro-algal habitats, and they're dominant grazers in these systems. Their major predators also live in both habitats, and that includes Pugettia bacillus and Lycosurius. In the case of Pugettia, it's an omnivore, so it eats both the plants that the snails live on and the snails themselves. With Lycosurius, it's a and so we had 7 different field sites which range from River Park down to Pilot Point. More specifically, we collected from False Bay, Mar Vista, and 4th of July. And so what did we want to do? We wanted to test the vulnerability of these two species of lacuna in both habitat types. And so first to do that, we had to determine the feeding rates of their predators on each species in each habitat. And so we did 4 specific things. And the first was to simply observe what they were feeding on in the field. The second was to find out their maximal feeding rates of the predators on the species. From then on, we wanted to see if there was an actual preference. And then from there, we said, okay, well, let's take this out, set up some mesopods, and find out the feeding rate from both habitat types. So feeding in the field, what did we do? In these sites, in the case of Pugettia, we went out, collected crabs, placed them into Ziploc bags with seawater, waited until they produced fecal pellets, and then took those back into the lab where we crushed them under microscopes. Crushed them under slides and viewed them under microscope, see what they're eating. And the great thing about Pugettia is that they're inefficient digesters, so we're actually able to see what they eat in the field. In most cases, when they do consume lacuna, you can actually see the shell in their feces. For Lycosurius, we collected the few that we could find and observed if they had anything in their mouths and in their arms and just considered that predation. So what have we found? Out of 23 crabs that we were able to collect feces from, we can see that all of them were feeding on microalgae and diatoms, whereas only 6 were feeding on lacuna. And when you look up here, these are what they look like or what the shells look like under the microscope. For Lycosurius, out of the 19 that we found, none were feeding on lacuna, some on barnacles or bryozoans, and most of them were not feeding on anything at all. Now, feeding in the lab, how did that work? Well, we measured the snails before and after in order to determine the size of the snails that were eaten. So for each trial, 10 snails were placed into a box with one single predator, and they were run for 3 hours such that the feeding rate was in number of snails given per 3 hours. And so what did we find? In our no-choice experiments where they were only given one species of lacuna to feed on, we found that the average number of things eaten was 2.8, and for variegata was 4.6. And through a G-test, we find that there is a significant difference in the number of variegata eaten compared to beans. However, the crabs were not size selective, and that was done through egg G-tests. Larger crabs did not eat larger snails, and that was done through regression analysis. Although we do have a p-value of less than 0.05 because of the slope, that's only 0.07, we did not consider that biologically significant. And to make things clear, on the x-axis you have carapace length of the crab, and on the y-axis is the size of the snails. Likewise, we did regression analysis to see if there's a relationship between the size of the crab and the number that they ate, and there was not. The regression analysis showed that there was not. Once again, the x-axis is edge fixed length and number of crabs on that. So for comparative experiments, we can see that when given a choice, instead of 10 single species, we had 5 of each species in each trial. We can see that they ate 2.7 for variegata and 1.4 for beta. And from the G-test, we see that there is a significant difference. So we can see that the proportion did not change from one experiment to the other, and that was in the G-test. So in other words, the feeding rates of the predators on each species did not increase, changing from single choice experiments to no choice. In the case of variegata, we see that the average is 1.9 for beta and 2 for variegata. The G-test revealed that there was no significant difference, and the stars were not size selective as well. Star size did not limit the size of snails that they preyed on, and that was under regression analysis. Once again, we do see a p-value less than 0.05. However, because of the slope, we did not consider that biologically significant. And there was no effect on the star size and the number of snails that were eaten. And for the preference experiments, where they were given a choice between beta and variegata, we see that they ate 0.5 for beta and 0.9 for variegata. However, through the G-test, we see that that was not physically significant. And once again, there was no difference in the proportion from one treatment, from one experiment type to another. So what do we find? We found that Pugettia ate significantly more variegata. Pugettia ate significantly more variegata than Leptostereus per unit time, and that was done through an unfair T-test. And the two predators did not have, they had the same feeding rates for. For our Oligocottus studies, which were the hardest to do, what did we do? We had 6 tanks, and each tank was separated into 2 arenas. The arenas look like this down here. And on one side, we had eelgrass, on the other, we had microalgae. In our case, we used Saccharina. And we placed 10 snails at the base of the plant and let them adjust for a few minutes. And then we added a predator. And these will run for 6 hours. And so we had 12 treatments, 2 species of snails, 2 predators, 2 habitat types, and the controls where there were no predators. And so we found that there was no significant difference in the number of snails recovered from either species between predator treatment, and controls, and habitat types. And so overall, the recovery rate of snails and controls was only from 8 to 10 seconds. So this graph, we can see if we pay attention to the control and the dashed line representing the loss rate, that 0 to 2 snails were never found in the control. And therefore, although we do see in the crab that they had a loss rate of 1 to 4 snails, we cannot conclude that that loss rate was predation because of our controls. Likewise, in our stars, we see that they did also have loss rates of 1 to 2. However, those were not concluded as predation because of our control. And while we did see some stars still had snails in their mouth when we collected, we could not do any scientific analysis on the root of it. And so Pugettia prefers variegata when given the choice, and they ate more in both the no choice and choice experiments, whereas Leptostereus had no preference. And neither predator had a was size selective, nor did its size affect the number and the size of snail that. So our next steps would be to basically improve the mesocosm studies by having more replicates and redefining the design of the overall tanks that we use. More data for more years on predators feeding in the field would be extremely helpful. And we know that leukemia population very consistently year to year. And lastly is having the historic work from Bruce Nagy, who actually showed that Leptostereus was an important predator in the field and showed that 15 to 50% of the sea stars in the field were actually found feeding on. And so my acknowledgments are the program, the University of Washington, Harvard, my very supportive mentor, Diana Padilla, Scott, Jessica, and then I'm big for being an awesome.
There's time for questions.
I saw Sasha was very fun. Me by simply observing and actually working really close with the snails. To me, the snail shell seemed a bit more fragile than the Linkage shell, and I can probably go along with that because I exited it out. So yeah, I think that it'd be easier for the crab to just kind of crush compared to the Linkage shell, in my opinion. But I can't really say that.
Did you notice maybe behavioral differences between the two lacuna species in terms of like does one hang out way up high and then the crabs have a hard time getting up there?
You're talking about the mesopausian experiment?
Well, actually, just in general, even running around in the tanks, because since your crab size didn't seem to impact whether they could eat larger snails or not. And so it seems like the snails are so small, any crab can eat any of those little snails. I was just wondering if their behavior changed, so something like one species would stay way up high.
Well, for the mesopausians, what we found when we went back to collect the snails after the trials were done is that the snails would actually go all the way to the top, and in some cases you can see them like on the side of the tank. So they knew there was a predator in there. They were like, I'm just gonna. So.
Can the crabs get up high too?
Yeah, the crab. Most of the cases, the crabs weren't in the. They were in the tank still, but they were like already outside the pods. But there were some cases where you could see them hanging from the tall grass where they were on the.
Do you think that it matters that crabs are visual predators and stars are not?
Yes, I do. I do.
Would that influence their preference?
True, yeah, sorry. Yes, I did not think about that, but you are 100% correct. Yeah. So, I think stars are just like they can get whatever they can get, whereas crabs are like, well, that might look tastier, so I'll take it.
All right, thank you very much. All right, the last talk of this first session is from Tess Dooley of College of Charleston, and her mentor is Tony Pires.
So I've spent the last 8 weeks working on the effect of pH on natural settlement and metamorphosis in an invasive gastropod. So most of you have likely heard of oceanification, but I'm going to walk us through the basics really quick. So through natural and anthropogenic processes, CO2 is emitted into the atmosphere, and some of the CO2 is then absorbed by the ocean, where it reacts with water to form carbonic acid. Carbonic acid then dissociates into a hydrogen ion and a bicarbonate ion, and the bicarbonate can then further dissociate into an additional hydrogen ion and carbonate. And as this graphic alludes to, this process is reversible, so the carbonate can react with the hydrogen ion to form bicarbonate. And bicarbonate and carbonate exist in equilibrium, and as you introduce more CO2, this is actually driven toward bicarbonate, so more carbonate is transformed into bicarbonate. And so the result is that with increasing CO2, we're seeing a decrease in pH of the ocean with an increase in hydrogen ions and also a decrease in carbonate availability as this transforms into bicarbonate. So for the past 10 years, there's been a lot of research on the effect of ocean acidification on marine organisms, and the classic example is that of the calcifying organism, because they use calcium carbonate to build their shells, and as carbonate becomes less available, it is harder for them to build and maintain their shells. But calcification is not the only process affected by ocean acidification. For example, studies have found that OA can impair the ability of larval clownfish to detect suitable habitat for settlement and also to avoid predators. For example, in a study by Monday et al., they found that when exposed to lower pH, larval clownfish it changed how they responded to different habitat cues. If you look at this graph with the cues on the x-axis and the percent of time spent in the water with that cue on the y-axis, you can see that they spent less time in the water with positive cues, which would be associated with good habitat, and more time in the water with cues associated with negative habitat. And this could lead to them settling increasingly in poor habitat. And what this tells us is that OA can affect chemosensory mechanisms and behavior. And if that is the case in larval clownfish, it could also be the case in other organisms. And that's what we were interested in. So a lot of marine invertebrates use chemical cues in order to induce a very important part of the life history, which is settlement and metamorphosis. Settlement is when larvae leave the water column and settle to the sediment. And this is usually followed by metamorphosis, where they transition from their larval stage into their ventive stage. And our study organism, marine Pugettia cornicata, uses cues from chemical cues from adults in order to induce this. And if you look at the adults, which form these stacks, you can kind of see why this might be the case. So like I said, they form stacks, and with largely sessile organisms, they'll stay in stacks for most of their life and reproduce within these stacks. So you can see how it would be advantageous for them to be able to find adult stacks on which to settle. And part of the reason we are really interested in this organism is that they've been extremely successful as invasive species. As you can see in this map, in blue, this is their native range from Nova Scotia to Florida on the East Coast. And in red, they've been found in the Pacific Northwest and throughout Europe and even recently in Japan. So fortunately, Pugettia cornicata have not found their way to Friday Harbor, but they can be found 4 hours south in the Tritonia Inlet, which is the southernmost part of the Puget Sound. And so in order to collect them, we had to drive down there at the low tide. When we got there, there's this mud flat, and you can see what looks like rocks in the distance. Upon closer inspection, it's thousands upon thousands of Pugettia, which I think is pretty good evidence for how successful they've been as invasives in this region. So we collected the individuals we needed and then headed back to the lab. So in the lab, we had to wait for larvae to be released. So Pugettia brood their larvae, which means that the females hold on to egg sacs until they develop to a certain stage, and then they release them all at once. So we left the adult in jars until they would release these larvae, and then we would collect the larvae. And at this stage, they are about 400 microliters, and as you can see, they're what we call villager larvae because they have these phelan. So we would put them into jars, and every other day we change their water and feed them single cellular alga crisis. And after about 2 weeks, they would reach between 900 and 1200 microliters. And at this point, they were ready for metamorphosis. So we did our metamorphosis assays in the Ocean Acidification Environmental Laboratory, which is housed in Lab 12. If you're not familiar with the setup, they have 16 tanks that can be set to a variety of pH and temperatures. And in these tanks, there's a constant flow through of filtered seawater, and pH is maintained by bubbling CO2 periodically through the water. In this lab, we had 3 treatments. We had 7.5, 7.7, and 8.0, all of which are pH that you would expect to see in the Tritonia Inlet during the reproductive cycle. And we kept the tanks at 20 degrees Celsius. In the tanks, we had 3-liter microcosms that were divided into 4 parts using modified jars that allowed for flow through. This flow through is maintained by using irrigation drippers that brought treated water from the tanks into these microcosms. In each of these jars, we placed a stack of individuals that were roughly the same size, and then we would add 20 larvae in them for a while. So after an hour, we would pull the container and we go out and check for individuals. And as you can see from this picture, the individuals were actually big enough to be visible on the adults, but we would look at them under the second microscope and collect them. And then we would score them as either larvae, which had to be in the lobes, or juveniles, which clearly lacked in the lobes. And while we were doing this, we were making 2 different measurements. The first was settlement, which we defined as all individuals, all the adults, when the adult was moved to a container, and metamorphosis, which was the complete loss of elim or transformation into juveniles. And while we were doing this, we were also taking pH and total alkalinity samples in order to describe the carbon chemistry of the water. So if you look at the first line, which has pH, you can see that we achieved our desired pH and that the pH didn't overlap between our different treatments. We also took total alkalinity samples, which are roughly similar for all groups. And from these 2 measurements, we calculated the PCO2, which is the concentration of carbon dioxide in water, and also the saturation states of calcite and ragonite. Calcite and ragonite are 2 species of carbonate, and any number greater than 1 indicates that they're supersaturated, any number less than 1 indicates that they're undersaturated. And as you can see, aragonite was undersaturated in the 7.5 treatment, and the larger shells of Opercular cornicata are made of aragonite, which means that in these conditions they could be dissolving. This shows our settlement data. On the x-axis we have our treatment, and on the y-axis we have the settlement, and each color indicates a different group, and we use each group for one trial, so we completed a total of 4 trials. And we did find that there was a significant effect of treatment on settlement. So settlement at 8.0 was lower than at 7.5. When you look at the different broods, the only brood in which treatment was significant within the brood was brood 9, but if you look at brood 12 in the gray and brood 16 in the white, you see a similar trend where settlement at 8.0 is lower or is equal to the other treatments. But brood 8 was the only one that showed an opposite trend. We saw similar results for metamorphosis, with 8.0 being significantly lower than the 7.7. And again, brood 9 was the only brood where this was significant within the brood. But again, there's a similar trend in brood 12 and 16, and kind of the opposite trend in brood 8. So where did this leave us? Going into this, I would have predicted that as you decrease pH, you'd see a decrease in settlement and metamorphosis. But we actually saw somewhat of the opposite trend, where settlement and metamorphosis were decreased at higher pH. But even though there was this decrease, they remained pretty robust across the pH range that we use, which, like I said earlier, is the pH you expect to see in their habitat here over the course of their reproductive period. And as estuarine species, we would expect that they'd see a lot of variation in pH in their environment. And so it makes sense that they have this tolerance to, seeming tolerance to a wide range of pH. And so going forward, it'd be interesting to kind of understand what the edges of that tolerance would be by testing a wider range of pH and also understanding the physiology behind this tolerance. And one other thing that we thought was really interesting is there's a lot of variability in response between broods. But because we only use broods once, we can't really determine if that variability is actual differences between broods or variation in between our trials that we can't understand. So hopefully in the spring I'll be working on answering this question by using broods more than one time to see if this response is with people over time. Thanks for listening and all the questions.
We have time for questions.
These snails scare the heck out of me. How did you keep them from sort of getting away and getting out?
They don't really move that much. Actually, so in the field, they don't really move that much. They stay on their stacks, but we found that in the lab, the males, which are the smaller ones with their cage sex, sometimes will move around. And we actually had one stack that kind of, they're usually stuck to a dead snail on the bottom, and one of them abandoned that and they kind of stuck to a different stack. And while we were taking them to freeze them yesterday, we noticed that it was a potted shape stack. But most of the time they don't move that much, so they're hard to.
Maybe if I could follow up, maybe I was worried about our quarantine.
Yes. I was just thinking that we might get in trouble if it turned out that these things suddenly appeared out there.
No, so we did have an ingenious solution to that, which was dumping everything that we used with them on the ground instead of sending it out into the water. So all of our waste was diverted to the ground where they would survive. So.
Cool. Because permits or no one would be happy if suddenly these things were out there.
If they show up,
So recent work on oyster larvae has shown that it's the alkalinity that's most important, not pH. So they're robust across pHs, but not to changes in alkalinity. So I was wondering if you thought about trying to manipulate the alkalinity to see if that would change, because your alkalinity was the same across all.
Well, since I didn't know that, but I think that actually really would be interesting to check out. Like I did see that in oyster larvae, they seem to have a difference in settlement or pH, but I think that would be an interesting thing to too, since we did have the same alkalinity.
Do you think that this particular species of Curbitula is highly unusual in terms of its ability to deal with pH compared to the other species of Curbitula that aren't as invasive as this particular one?
I don't know the answer to that, but I do think that this particular species has been a lot more successful as invasive, so that could be good information. And since you find a lot of these other particular species in the same range, you can test their tolerance in comparison. But I don't know.
Any more questions?
So you said the stacks, they form these stacks, right? So is there like some kind of mineralization in the stacks?
Well, usually we don't know exact sex, but they change sex at some point. So typically when they settle, they're male, and the existing ones, the bigger ones, are females. But they have found that if one settles not on a stack, it will automatically be female. So I think there's typically less males on a stack. I don't know.
Yeah, okay, great. Yeah, following up on Diana's question. It turns out that you can grow digital larvae at pH 7.2 in instant ocean, which has really screwy carbonate chemistry, and in fact is artificially buffered at really high alkalinity. So what would you expect might be true then about but what else would you want to look at then? So we've got pH, we've got alkalinity, and what else about the carbonate situation would you want to look at then? Would you want to manipulate pH, you can manipulate alkalinity, what else do you think? Like the sort of towards the bottom of the table.
Oh, the saturation states of the yeah. I mean, we did see that it was, it was undersaturated for that one, but we didn't actually see an effect in that group. But I would, I guess that's not really that.
No, just suggesting that there's different ways to manipulate carbonate saturation.
Yeah.
You could do pH.
Yeah.
All right, thanks a lot. So thus ends the first session. Please join us at 7 o'clock.